Okay, so today in this video we're gonna discuss about lysogenic cycle. So as I gave you the we can say overview in the last video, so lysogenic cycle is basically what it includes lambda phases. It includes lambda phases, and now lambda phases are those phases which have less developed fibers or no fibers. Okay, which contain less developed fibers. So now remember in this, like the bacteria in which the lysogenic cycle take place are known as lysogenic bacteria. Okay, lysogenic bacteria in which lysogenic cycle take place. Okay, now in this cycle, virus infect bacteria without killing them. We know these are basically in this cycle. What actually will happen? Virus will infect the bacteria without killing. Infect the bacteria without killing. Okay, without killing. Now here we have several steps. If I'm going to talk about the first step, so that is step 1, which is absorption. Okay, so what will happen in this step basically, like we have first the genome will be absorbed, matlab attachment. Here we have this bacteria, here bacteria genome is present, and here we have bacteriophage which will attach to the bacteria. So this is the first step, which is attachment, absorption, attachment. Okay, so now second we have our genome penetration. Genome penetration. So what will occur here in this genome penetration? What will occur? The bacterial genome will penetrate. So for suppose this is bacteria. This is the bacterial genome, and here we have bacteriophage. And the bacteriophage means genome is penetrated. Now here you can see the genome of bacteriophage. Fine. Now remember this. This genome penetration is also known as latent stage or latent period. Latent stage or latent period. Fine. Now we should know that in which phase DNA just enter the bacteria and do nothing. Like in the cycle, what will happen? The bacteriophage DNA is entered here. Now it will enter and it will do nothing. It will enter, but it will do nothing. Okay, what does this means basically? This means like that is in the prophase or temperate phase, in which bacteria enter and do nothing. That is known as we can say prophase. Uh, okay, prophase or temperate phase. Now we should go with the gene. We can say regulation. I may explain you because as we have latent stage, bacteria is entered and do nothing. So that's the about gene regulation. Okay, so what will happen in the gene regulation basically? So like gene activities, basically the gene regulation, there will be gene activity which is turned off. So here phage of, we can say bacteriophage DNA genome activity, or we can say bacteriophage DNA entered and it will do nothing. Why it will do nothing? Because this DNA activity is turned off. This DNA activity is turned off. Now it doesn't mean it can never on. No. Just because of some unfavorable conditions, like we can say it includes unfavorable conditions, like you can say UV radiations, or you can say mm, it will turn on phage and lactic cycle start. Like some unfavorable condition like UV radiation can occur, and when these UV radiations or some different unfavorable conditions will occur, so what will happen? So gene activity will be turned on, and when it activity will be turned on, so again here lactic cycle will start. Lactic cycle will start. Okay, so now let me tell you how. So with the help of diagram, may I explain you. So yep, first of all here we have absorption. This is the bacteria DNA. This is bacteriophage. Okay, so this is attachment or absorption, you can say. Now we are going with the second, which is genome penetration. This is the bacteria. This is the phage, and the phage we can say here we have phage genome. Now phage genome is entered here. So here we have phage genome. Okay. Now third process will occur, that is integration. Now what will happen in integration? Now the phage is conjugated or interacted with host DNA. जो phage होगा bacteriophage वो क्या होगा, connect हो जाएगा किससे, host के DNA से. For example, this is the host DNA. Now this DNA will be connected to the host DNA. Okay, now here this is the bacteriophage DNA. So this process is known as integration. Okay, integration. Now we are moving towards replication. As we can say, this is conjuncted. It is also known as prophage. Its host genome here is known as prophage. So now let's continue it furthermore. Yep, now we have fourth process, that is replication. Now if this is done, now it will divide to replicate. It will replicate like this. Okay, so when it is replicated, so now here if I can write here the process of replication. Now this is done. This replication is done by binary fission. Binary fission. Okay, so when this is done, now if we can say unfavorable conditions occur, which means if we can say UV rays can infect, so this means on that time the lactic cycle will start. So here what, here lactic cycle will start. This means it will start the synthesis of endonuclease, and which will destroy the bacteria. We can say it will destroy the bacteria. Okay, so this is how the lysogenic cycle and lactic cycles are. Now in the next video I'll tell you difference between lactic and lysogenic cycle. Till then, bye bye.